position in the bioinformatics group of Andrew Page at the Quadram Institute of Bioscience, where she is a member of the Gut, Microbes, and Health Institute Strategic Program. She graduated with a PhD in 2019 at the Max Planck Institute for Marine Microbiology in Bremen, Germany, where she focused on an endosymbiosis between bacteria and bivalves in the deep sea. Her work has been mostly metagenomics towards teasing apart highly similar strains of bacterial symbionts within a host. One interesting fact is that during her PhD, she participated in 2 research expeditions to the hydrothermal vents and cold seeps. She is fascinated by host microbe interactions and how this links to strain diversity. Please everyone signal your applause to welcome Rebecca. Take it away.
Thank you very much, Lee. And before I start, can you hear me? Sorry.
Yes.
Great. So before I start, I just want to thank you, Lee, and Nabil for organizing this excellent conference, which has been a lot of fun, and I learned a lot, as this is a very new community to me. So yeah, that has been great. And I also hope that you are not yet in your evening and you're still with me and up for a little adventure from infectious disease to the deep sea. And as my title already kind of gives away, I will be looking at what lies beyond 16S rRNA sequences. But already the other 2 talks before have been showing why this is highly relevant. Just to introduce a little bit the topic, I just want to introduce what usually you would be looking at if you go into a natural environment that you don't know yet, you don't know what you're going to find. The first thing that you're usually interested in, if you get a sample from the ocean, if you get a sample from soil, or also of any kind of host, human or animal host, the first question that is usually coming up is how diverse is the bacterial community in the natural environment. And obviously this question extends to, first of all, that we want to know what is the community composition like in a particular environment, for example, in a host. And what lies basically beyond that question is that we really want to understand who can coexist and how do they do that. Traditionally or typically, the first thing in a new environment, such as a human gut microbiome, for example, would be that you do 16S rRNA sequencing, and you get one of these bar plot profiles here that basically show you the community composition and maybe abundances of the different members of the community. However, it gets really tricky if the 16S rRNA sequence doesn't provide the resolution that we really need in order to understand the community that we are trying to understand. And this is where strain diversity comes in. And I mean, already both, or especially the first talk today, has been showing why understanding strain diversity in the first place is very relevant in basically all different kinds of research areas when we deal with microbes. Just a little warning, if I'm talking about strain throughout my talk and also in the project that I'm talking about, I usually refer to a bacterial strain if it is a bacterial genotype that differs in at least one nucleotide from another genotype of the same species. And I'm aware that there are different ways of describing different strains, but in this project we are dealing with unculturable microbes, and so one way to distinguish strains from each other would be nucleotide polymorphisms. And in different kind of systems, and since I'm coming from an environmental point of view, I'm showing you this beautiful Vibrio squid symbiosis here as an example, where a single nucleotide polymorphism in a regulatory gene can already impact the ability of a microbe to colonize its host. However, I'm not working with squids, but I'm working with these beautiful animals that you see in the screen here. And this is a deep sea mussel of the genus Bathymodialus that lives a couple of thousand meters deep on the ocean floor. These animals basically dominate hydrothermal vents and cold seeps throughout the oceans. And you can see here basically a map of all of the hydrothermal venting systems that we know up till today. So they really dominate all these spreading centers that you have on the ocean seafloor. And in the 1970s, these were basically discovered, and to the scientists' surprise, these environments were basically full of life, full of animal life, which was completely unexpected, as usually the deep sea is considered a desert where you don't have any sunlight and no food from the surface areas. And the reason why this is so covered and dominated by so many different types of animals is that these animals have established very tight and intimate symbiosis with microbes that can use this chemical energy instead of sunlight energy in order to produce biomass and sugars that feed the animal hosts and sustain the life at these vents. This is basically the system that I've been studying, so the bathymedailous muscle. And if you open one of these animals, the first thing you see is that the gills, the gill organ, indicated here in yellow, is highly enlarged if you compare it to a shallow water muscle, for example, the blue muscle. And the reason that this is enlarged is that the epithelial cells within the gills is packed with intracellular bacteria, which use this chemical energy at the vent sites in order to produce biomass and feed their host. Typically, the bathymedailous muscles have only two types of symbionts that they're primarily associated with. One is a sulfur oxidizing symbiont, or SOX symbiont, that uses reduced sulfur compounds at the hydrothermal vent in order to fix inorganic CO2 and makes biomass out of it. And a second phylotype, a methane oxidizing symbiont that uses methane instead to produce biomass. These symbionts are usually transmitted horizontally from one host generation to the next, which means that every new host generation has to take up the symbionts from the surrounding seawater and gets colonized by it de novo every generation. In the project that I have been focusing on, I mainly work with the sulfur oxidizing symbiont that you can see here. And as I said already before, the muscles are usually colonized by a single highly abundant 16S rRNA phylotype of the sulfur oxidizing symbiont. So if we were doing 16S RNA sequencing on those muscles, my talk would be finished here because that's already the final result. So obviously, we were interested in strain diversity in these systems. And not only we wanted to know whether there is strain diversity, but we also wanted to know whether this has any implications for the symbiosis, whether this strain diversity really matters for the metaorganism. So what we did was we went to the Mid-Atlantic Ridge, and you can see that all my sampling spots are here in the middle of nowhere, which is in the contrary to all the other talks that we have seen that had always sampling spots on either of these continents. So we went there with the ship for a couple of weeks and used remotely operated vehicles to dive deep down into the ocean to then collect the mussels from the seafloor from the hydrothermal venting systems. So we collected animals from 4 different hydrothermal vent sites, and in total we included 18 host individuals in this study. What then I did was basically for each of those vent sites, we sequenced the metagenomes of the symbiont carrying organ, the gills, and I took all the metagenomic reads, combined all the reads from one single site, and did a co-assembly of the metagenomes. And then I performed some binding steps that basically grouped metagenomic assembled genomes of every SOX symbiont for each site separately. So this resulted in one reference sequence for the SOX symbiont for each of those sites. So the first question was, do we have strain diversity? So do we have any kind of heterogeneity within the population of single host individuals? And to answer these questions, we performed a SNP calling approach where we used this co-assembly per site as a reference sequence, and then we mapped all the metagenomic reads for each muscle separately to this co-assembly, and then we called the different SNPs in order to understand how heterogeneous is the per muscle population, so to say. And before I show you the results, I just want to put you a little bit into perspective what we would usually expect from an intracellular symbiosis from other studies that have been doing similar kinds of analysis using SNP data. So you can see here other symbioses such as aphid symbionts or rift-ear tube worms that you have also colonizing the hydrothermal vents in the deep sea. You have Solemaya clams that also have intracellular symbionts. And you can see that usually these nucleotide polymorphisms that you find in the symbiont community within the host is really low. Of course, if you go to the other end of the spectrum, to the human gut microbiome, where you have a lot of different species, not intracellular mostly, but extracellular, you also have quite a range of heterogeneity within each of those species, which is much higher than in these intracellular symbiosis that you see here. So when we looked at our data, we were pretty surprised that the heterogeneity that we find within single host individuals was actually much more similar to the human gut microbiome than it was to other intracellular symbionts. So we did actually see quite some substantial symbiont strain diversity beyond identical 16S sequences within those single muscles. So having this information, having this diversity at within muscle population level, we could actually now start to answer a lot of different other kind of questions. And this is great for this particular system because first of all, we cannot culture any of the symbionts in the laboratory. And second, even keeping the muscles in the lab alive is quite a challenge and doesn't work for all muscle species. So this basically helps us to complement for the lack of culturability for these symbionts at this present stage. So using the SNP data, we really wanted to understand how similar now is the symbiont population of two different host muscles that occur at the same site. And to answer this question, we use a different kind of measure called FST. And it's very simplified. FST basically tells us how similar two different population of microbes are that you are comparing. So if it's very high, so closest to 1, you have very dissimilar microbial populations in terms of abundance and presence of the different strains. And when you have very low FST, this means that your populations are more similar to each other. So again, I'm using the human gut microbiome study that I showed before from Schleusnig et al. As a comparison. So here you see basically how similar two different host individuals were to each other when you compare the human gut microbiome at the strain level. If we do the same thing now for the muscles, and if you remember, we had a rather similar degree of heterogeneity in those, we do see that all the FST values fall within this red highlighted area, which basically tells us that the symbiont populations of co-occurring muscle individuals are rather similar to each other. And we can really learn quite a lot from that. For once, we can actually now have an idea of how the symbiont transition works, because this very low or very high similarity between the symbiont population is an indication that the muscle hosts throughout their lifetime continue to exchange the symbiont community with each other and with the environment. So this tells us a little bit about the colonization dynamics that we have in this particular symbiosis. We also wanted to know how many symbiont strains can actually coexist in a single host. If you cannot culture the symbionts, this is a quite tricky question to answer. So we again turned to the nucleotide polymorphism data that we had, and using SNP linkage and SNP frequency approach, we basically took all the genes that are present in every single bacterial cell, so that have an even coverage, and I come to that in a bit. Counted or reconstructed and counted the number of different sequence versions for each of those genes. And then we took the genes that had the highest counts of sequence versions as a proxy for how many strains we have in a single host. Of course, for all this comparison, we usually always take the same read coverage of 100x to be able to compare between host individuals. And by using that approach, we identified between 6 and 9 strains to be present within single muscles. And keep in mind that these are intracellular symbionts, which is something that is not really expected to exist stably in a mutualistic symbiosis. However, when we took also the libraries that had a higher read coverage and used that higher read coverage with the same type of analysis, we saw that we could increase our sensitivity and detect even more strains within single host individuals. And we detected up to a maximum of 16 strains within a single host. So of course this was coverage dependent, and if we had higher coverage, we might even detect more strains in there. So this is up for future studies to see. So now you might ask, okay, you have all the different kind of strains in there, you have nucleotide polymorphisms, but does it do any, does it have any effect on the symbiosis? Does it have any functionality? And of course, we cannot test any phenotypic implications of those SNPs in the laboratory on these bacteria. But we still wanted to know whether these different coexisting strains have a functional differences in the symbiosis or for the symbiosis. So we came up with another way to look at it. Again, we used our metagenomic data. And basically here you have on top, you can imagine the reference sequence or the reference context of the core assemblies. And then we again mapped all the metagenomic reads to this reference sequence. And then if you look at some of these contexts, you see that sometimes you have coverage drops, as in these regions here, where the reads mapping to this area is much less than in the rest of the context. And this made us think that actually these kind of regions of low coverage represent genome fragments that are not encoded in every bacterial cell, but just in a fraction of the population. So we made use of that information and screened systematically the entire metagenomes per muscle for these low coverage regions. How we did that was we first took all the single copy marker genes where we know that every single bacterial cell of this bacterial group has to encode these and has to encode these only once. So we know that the range of coverage that we find for these genes is in the normal way of how a single gene that is present in every cell should be covered. And then we took every gene that we find with coverage values below this and considered this as potentially strain-specific or low coverage. And then, of course, we had to also apply a couple of filtering steps, for example, excluding all these low coverage genes identified in contact edges, which is usually where the assembly breaks. And so often you have some weird kind of repeat structures there, but also in the end of context, you usually have this coverage drop, which is due to the mapping. So that's why we excluded everything that fell into contact edges for this analysis. And in fact, we did identify in some of the host individuals even up to 50% of the gene content to be part of this strain-specific or low-coverage gene pool, which was quite impressive for us. However, most of these genes cannot be annotated, so they were hypothetical proteins, so we still don't really know what they're doing. But there were a few genes that could be annotated functionally. And first of all, you have the usual suspects, right? You have the phage defense mechanisms such as CRISPR-Cas, you have the surface structures like O antigen, and in the first talk today we heard quite a little bit about those. So we saw that there are differences in the gene content which are responsible for producing these. But we also found different strain-specific genes in, for example, phosphate-dependent gene regulatory mechanisms, and also even in key metabolic genes that play an essential role in the symbiosis, as these are pathways that allow the bacteria, for example, to use additional energy sources such as hydrogen in order to do their carbon fixation pathway, which is the key function of the symbiont for the symbiosis. So we were quite surprised that we found so many different important mechanisms to be strain-specific. And we found that how abundant a pathway was, in some cases dependent on the concentrations of these compounds in the environment. So there were differences between the different sites that we had. which made us think that this might be a very good adaptation and very flexible mechanism to have different strains encoding different functions that then they can adjust the abundances in the metaorganism. We had some transcriptomic data as well that confirmed that most of these pathways that we find to be strain specific were expressed, which suggested that probably some of these pathways really do play a role also phenotypically in the system. Now, for all of you who are not so much into the bioinformatic part of the analysis, I have some visual treats, which unfortunately is not my work. But it's the work of Miguel Angel Gonzalez Porras, who has developed this fluorescence in situ hybridization approach, or gene fish, where you label single genes or gene clusters within the natural tissue, basically. And he has really mastered this method. And what he did, you can see here the single gill filaments, and the symbionts are labeled in green. So the 16S RNA, which all of them have exactly the same, is labeled in green. But then you see two of the genes that we discovered with the low coverage analysis, he labeled in two different colors. And you can see that we have some bacteriocytes, so these are the host cells basically, some bacteriocytes that are carry, for example, only the strain that can use hydrogen as an additional energy source. And then you have, for example, here a strain that cannot do that. And here you have a strain that has another gene function. So in one picture, you already see the distribution of at least 3 different strains in the natural gill tissue. So this was a really nice confirmation of our bioinformatic analysis. And very impressive to see. So just to wrap this up, using all the metagenomic analysis and an uncultured organism, we could actually understand quite a lot about colonization dynamics. We saw that the Bathymodalis mussels likely exchange their symbionts throughout their lifetime with the environment and or with other hosts. We could detect up to 16 symbiont strains within single mussels. And we saw that these strains differ functionally from each other in their functional capabilities. All of these together led us to a hypothesis that the strain diversity and maybe even the competition among these strains in the system, instead of being a disadvantage, which is usually assumed for intracellular symbiosis, instead of being a disadvantage, this may actually be an advantage and further optimize metabolic productivity of the entire population. If, like in this symbiosis, if the environment and not the host feeds the symbiont. So the symbiont doesn't really have any chance to very much exploit the host as it gets the environmental energy. And this, of course, can also apply to other symbioses that have similar features, for example, coral symbiosis, etc. And now, just to wrap this up with a couple of general statements, I think that even though this is a very remote symbiosis somewhere in the deep sea, I think the topic of strain diversity within natural communities is very relevant. As I said in the beginning, even a single mutation can make the difference, a functional difference even. And by looking at these fine-scale differences, this really helps us in understanding the population dynamics and colonization of communities where we cannot yet culture the organisms. Pangenomes can be large, even if you have identical 16S sequences. And then I also think if we go to a natural community, we are always very fixed with the idea that we want to have a precise species affiliation of the community. But actually, I think that sometimes it might just be more valuable to mainly look at the functional differences that we find there instead of going always after the precise species affiliation. Yeah, and I think this brings me to a huge thank you to everyone involved in the study. This is the previous group where I worked in the lab of Professor Nicole Dubillier, all the co-authors of the study. Of course, I also want to give an acknowledgement 2.0 to the new group where I started a couple of months back, especially to Andrew and Andrea, who have been really great. Yeah, I'm happy to answer any questions if you are still awake.
Thanks so much. You definitely have some questions here.
Great.
All right, and pardon me again one more time. I'm scrolling at my phone. All right, so the first question actually asked was, do you yourself actually dive in a vessel to those depths in the middle of the ocean?
I wish. Unfortunately, so I went to two of those expeditions, and we had remotely operated vehicles, which are kind of robots. So you sit in a container on the ship, and you have a lot of screens, and then you have some really skilled pilots, which I'm sure were great at video games when they were younger, that operate this robot arms in the deep sea. So I didn't dive down, but I saw at least live the hydrothermal vents on the screen.
Okay. Maybe we can hear about the tales of you diving down next time. The next question was, do you have any plans of looking like the host diversity of where you get these symbionts?
Yes, so colleagues of mine are actually looking at that. They're looking at, some of these sites that we see in the Mid-Atlantic Ridge also have hybrid zones where a couple of different host species hybridize, which is also very interesting to understand host symbiont dynamics. So they're definitely looking at that, yeah.
Okay, wow. And then at least a couple people caught an interesting slide where you had some names on there: Lucky Strike, Clueless, some other names. Were those names of like the metagenomic samples, or what? And also, what was the naming scheme?
Yeah. Oh, sorry. I skipped that. So if you are going on one of these research expeditions and you're on the ocean for 5 weeks, and you don't find any vent, and suddenly you find the vent you were looking for, you call it Lucky Strike or Clueless because you don't have any idea where you are. So these are usually the names of the sites. All these dives are often done late at night, etc. So scientists crank together, drinking a lot, come up with these kind of names.
That's funny.
Yeah.
And then this question, this question is, were the SNPs defined by mapping? And the person who asked the question says, I'm wondering about the jump from 100x to 370x. It seems too much.
Yeah. Okay, so I think I didn't, I did not a great job explaining that. So so we mapped all the reads to the reference, and for all the comparative things, so the SNP calling and all these kind of things, we basically downsampled. So we always had an average coverage of 100x for every sample. That's what we did for all the comparative things. And then when we tried to estimate the number of strains, we also did that for all, like for this average 100x coverage. But then we were just wondering, what if we use the more sequencing data that we had to try to see whether the number of strains we can estimate increases? And that's exactly what we did. So the highest coverage we had in example was 370 something. So we just looked using the full coverage, how many strains we can estimate from that. And that's where we found the 16. Yeah.
That makes sense. And a follow-up question was there a filter for small genes in the gene or strain analysis?
For the low coverage analysis?
I believe so, if
Yeah.
I think it's in the context of that.
No, we did not really filter by gene length. So we did I think I see where this question comes from, because so what we did was we did still the mapping to the entire context, and then the only thing we did is just extract the gene, so the open reading frame basically, and extract the coverage values from there. Because if you map to each gene separately, you have these coverage drops, of course, in the edges, and then gene length makes a huge difference if you just count the number of reads. So that's why we did not do that. That's why we need this consensus assembly where we mapped all the reads to and then extracted the genes. But I'm happy if I didn't answer the question correctly, please let me know, and then I can try to do a better job.
Sure, and you'll see that question on Discord and any other follow-up conversation. I've actually lost track of the entire conversation myself, so I think everyone please thank Rebecca. We're going to continue the conversation online for our happy hour.